In this experiment, you'll study how electrical signals are transmitted along a simulated nerve fiber, such as an axon in the human body. This plastic straw has been filled with a mixture of agar, which is a plant-based gel, and salt, which allows the gel to be mildly conductive. Our signal will be sent by this 9-volt battery. First, let's study the conductivity of our nerve fiber. These short lengths of steel wire act as electrodes which can be inserted directly into the agar gel. To determine the conductivity, we must measure the cumulative resistance at different points along the axon's length. To find the resistance, we will measure the current and the voltage and use Ohm's law. The battery will be connected to both ends of the axon, giving us a constant current throughout the length of the axon. To measure this current, first connect the multimeter in series with the axon and the battery. Now we connect one end of the multimeter to the positive terminal of the battery, and the other to this needle probe that we can stick into the plastic straw at different points along its length. You can see that the voltage difference between the positive end of the axon and the needle probe increases as you move along the length of the axon, showing that the resistance increases as well. By measuring the cross-sectional area of the axon and using the measured resistances and lengths, the resistivity of our simulated axon can be determined. Conductivity of a material is just the inverse of its resistivity. Next, we will simulate the demyelination of a nerve fiber. Demyelination occurs when the protective outer sheath of a nerve fiber is degraded, which causes some of the energy of electrical signals to be lost into surrounding tissues. This tray of salted agar will represent the surrounding tissue in which our nerve fiber is embedded. Poke some additional lengths of steel wire into the axon. This will simulate areas where the nerve fiber has been demyelinated. Insert the other end of the pins into the tray of agar. Now measure the voltage difference between the positive end of the axon and different points along the length of the axon again. You'll see that the voltage difference increases over a much shorter distance, showing that the voltage level of a nerve signal could drop below the action potential before it's able to be retransmitted.